medical immunology is a rapidly advancing branch of medicine. The exponential growth of immunology during the second decade of the 21st century has its own implications for medical professionals and USMLE and COMLEX test designers in particular. The major issue of concern pertains to the identification of concepts that are relevant to physicians' roles as healthcare providers and patient educators, and to distinguish them from those concepts that are only relevant to the science of immunology. In preparation for your board exams, we must bear in mind that the purpose of USMLE and COMLEX examinations are to test medical students on concepts that are applicable to enhancing their knowledge to practice as physicians in the United States. Throughout the history of medicine, anatomy and surgery represented the core of the medical field. Within recent years, biochemistry has assumed this fundamental role. Now, looking to the future, immunology may dominate as the field that progressively defines the spirit of futuristic medicine. This online course and its associated workbook are one of my latest scholarly products in medical board test preparation. The course was created to be highly exam relevant, integrated, and clinically oriented for you. There is a lot of information to cover, and I will focus on immunological concepts that are high yield for your examination. As we go on and cover more material, we may revisit some of the earlier concepts from a more immunologically sophisticated perspective. So, if you are ready now, we can begin the show. First and foremost, let us describe the immune system. Let us do some definitional work. Immune system is a system which is designed to hurt the pathogens, that is, to hurt the non-self and to leave the host or the self intact. There are other definitions as well. For instance, one functional description is that immune system is a system of many biological structures and processes that protects organisms against diseases. Another functional description is that this is a system that detects a wide variety of agents, from viruses to parasitic worms, and distinguishes them from the organism's own healthy tissues. A categorical description would be that this is a system composed of two subsystems, the innate and the adaptive immune systems. And finally, an anatomical description would be that this is a system with two arms of humoral and cell-mediated immunity. Before I go further and of historical perspective, it is important to talk a little about the concept of immune privilege. Just to know, some of the organs in a body are shown or have shown that they can tolerate introduction of antigens better than other tissues without producing an inflammatory immune response. It has been a historical observation that tissue grafts are normally recognized as foreign antigens by the body and they are attacked by the immune system. But it is shown that if they are grafted into privileged sites, they tend to survive for longer period without being rejected. So there is something about these organs or tissues that makes them less susceptible to inflammatory responses. Traditionally, the privileged sites would have included brain, eyes, placenta and fetus, testicles, and articular cartilages. So the question that we're going to ask ourselves is that what is the purpose of immune privilege? We have an evolutionary sort of explanation for it. So evolutionary wise, we do believe that this is an adaptation to protect vital structures from potentially damaging effects of an inflammatory immune response. Of course, the current trends tend to somehow disagree with these historical trends. For instance, initially CNS immune privilege was believed to be due to its isolation from immune system by the blood-brain barrier and the lack of draining lymphatics. However, lately, the obvious presence of immune-competent microglia in the CNS and certain autoimmune CNS diseases and neurodegenerative conditions such as Parkinson's, multiple sclerosis, and Alzheimer's has challenged the original views. With recent developments in neuroimmunology, it is now believed that CNS is not fully isolated from the peripheral immune cells. Furthermore, it is shown that meningeal spaces include both innate and adaptive immune cells that use deep cervical lymph nodes as their portal of entry into the meninges. Before I go any further, it's important to know what we are talking about, and to be precise for communications hereafter, it's very important to define some important immunological terms. So first and foremost, I like to define immunogens, antigens, epitopes, and haptens. And we like to see how we can distinguish these from each other. Immunogen is any agent capable of inducing an immune response, whether it is humoral or cell-mediated. Antigen is a molecule which can be bound by an antibody, or by a B-cell receptor, or by a T-cell receptor. Epitope, or antigenic determinant, or antigenic specificity, is the minimum molecular structure on an antigen which binds to a specific antibody molecule or to a T-cell receptor, or can be presented on an MHC complex. We're going to talk about MHC complex shortly. MHC stands for Major Histocompatibility Complex. To illustrate an epitope and distinguish it from an antigen, this diagram I feel is very helpful for you. A particular antigen molecule may have several different epitopes or determinants. Each one of them is capable of binding to a specific antibody. For instance, in this particular antigen that I'm showing here, we have 4 epitopes, and each one of those epitopes is able to bind to a specific antibody produced by the plasma cells. So what is the hapten then? Hapten is a low molecular weight molecule that binds to antibodies or to T-cell receptors, but it cannot initiate an immune response in isolation and alone unless it is conjugated to a suitable carrier molecule, which is usually a protein. To summarize, all immunogens are antigens, but not all antigens are immunogens. And again, to summarize, haptens are antigens because they can be bound by antibodies, but they are not immunogens as they cannot induce immune response without a carrier molecule or carrier protein. let's talk a little about innate and adaptive immune system. I'm gonna touch base with this a little later as we learn a little more about the basics that we have to know about the immune system. So first and foremost, what is the innate immune system? This system is the more primal of the two systems. It is available at birth and provides broad and non-specific protection against the pathogens. This is the system that even the lower organisms have it. Immune cells of the innate system are neutrophils, macrophages, dendritic cells, and natural killer cells. So these are the characters that we're going to be looking at a little down the road as we're going to get more specific about the function of the innate immune system. These innate cells have receptors that identify broad classes of antigens and pathogens that are specific to all major pathogens, such as gram-positive and gram-negative bacteria, RNA or DNA viruses, fungi, or parasites. By this I mean, for a neutrophil, for a macrophage, it doesn't matter. The moment they see essentially the patterns that are specific to all viruses, they react similarly. So they react similarly, let's say, to all DNA viruses. So they have a more of a broad and generic type of response and function. After activation and recognition of generalized pathogenic patterns by these cells, they produce cytokines and enzymes that, in addition to the pathogens, quite often may also damage the host cells and tissues, and as we see later, quite often may cause the death of these cells as well. So let us summarize the components of the innate and adaptive system and get a feel for how they are differentiated from each other. With regard to the cells, the innate cells include the phagocytes, such as macrophages, and monocytes are practically the precursors of macrophages in the bloodstream. Macrophages are in the tissues, monocytes are in the bloodstream, and as they migrate to tissues, they are converted onto macrophages. Then we have neutrophils and APCs. APC stands for antigen presenting cells. The most prominent APC cells are dendritic cells, but we have more. The other innate cells are mast cells and basophils, eosinophils, and natural killer cells. In contrast, the cells of the adaptive system include cytotoxic T cells, T helper cells, B cells, and plasma cells that, as we're going to see later, are derivatives of B cells, antigen-presenting cells that link the two systems together. I have a little bit more to talk about linking APC cells. Then we have secretory products of innate and adaptive system. For the innate system, these mainly include complements, cytokines, acute phase proteins, interferons, and lysozyme. For adaptive system, these are antibodies and cytokines. Let us contrast a little bit more closely the functional and anatomical differences of the two systems. With regard to innate system, it provides early, immediate, short-term, with maximal response to the pathogens. And this response does not change. Response of the innate cells do not change with further exposures. They are maximal from the beginning. In contrast, with adaptive system, this system provides with a delayed and long-term responses that are progressively getting stronger and more refined with further exposures. And I want you to know that it takes at least about 3 days for the first response of adaptive system to happen, whereas the innate response is almost immediate. Innate system recognizes a limited number of molecular structures and broad classes of the pathogens. It just, for instance, recognizes the RNA viruses or DNA viruses or bacteria. It cannot tell if that gram-negative bacteria is a Klebsiella or is an E. coli, for instance. The adaptive system, in contrast, recognizes a wide variety of molecular structures and it identifies specific antigens. Again, innate system provides generic responses. It has conventional and fixed genes. Another term for this is germline encoded. It has a broad antigenic response. For example, innate system has genes for proteins to identify broad bacterial motifs or characteristics, if you wish, such as lipopolysaccharide, LPS, peptidoglycans, or bacterial flagellum. In contrast, adaptive system provides for a specific response. It has unconventional genes. It has antigenic specificity. For example, it has genes that adapt themselves to a specific bacterial antigens on a specific, let's say, gram-negative bacteria, such as Klebsiella. Innate system receptors have genomic specificity and do not require gene rearrangements. A given set of receptors are present on the same cell types. For instance, all neutrophils have the same set of receptors. In contrast, adaptive system has receptors like T-cell receptors that they lack genomic specificity and they require gene rearrangements. And similar cell types, let's say T cells, they express different receptors on their surfaces. Receptors of innate cells are similarly expressed by similar cells, as we said earlier, like by all macrophages or all neutrophils. Receptors of adaptive cells, such as B cells or T cells, are differently expressed by similar cells. Innate cells, they are short-lived, whereas the adaptive cells, they have a longer life. And as we're going to see later, some of them, they live as long as the individual lives. Location-wise, innate cells are found almost anywhere in the body, but mainly they are found in places that pathogens may enter first into the body, such as under the skin, on mucosal surfaces in the GI, lung, and urogenital tract. Adaptive cells are mainly confined to the bone marrow, to thymus, spleen, lymph nodes, lymphatic vessels, and blood. Let's go quickly over this diagram so that you can get a little oriented about the cell types of adaptive and innate system and trace their origin. They all get originated in the bone marrow from a so-called multipotent stem cells. These multipotent stem cells, they have the potency to produce lymphoid stem cells and myeloid stem cells. Lymphoid stem cells, they can migrate to thymus, and in the thymus, they're going to produce the T lymphocytes. And T lymphocytes can appear in the blood and also in the tissues. By the same token, from the lymphoid stem cells, also in the bone marrow, we produce the B cell precursors. These B cell precursors, in the form of B lymphocyte, can appear in the blood, and later, as we see, B lymphocytes can change into plasma cells in the tissues of the body. Myeloid stem cells in the bone marrow can produce granulocytes and pro-monocytes, megakaryocytes, and pro-erythrocytes. Products of granulocytes and pro-monocytes in the blood include monocytes, neutrophils, eosinophils, and basophils. Monocytes, when they migrate to tissues, they turn into macrophages. So monocytes in the blood, macrophages in tissues. As you see here, I have mast cells in the tissues, and I have a question mark to show that they are derivatives of the basophils, possibly. So there is a question mark in there, means that it is questionable. Are the mast cells derivatives of basophils or not? But functionally and anatomically, they are closest to the basophils. Megakaryocytes, as the name implies, the mega means that these are big cells, but as they evolve in the bone marrow, they get smaller and smaller, and they turn into platelets in the blood. And of course, pro-erythrocytes, they make the red blood cells. This is another diagram that shows to you how these cells look in most of the diagrams and most of the pictures. The multipotent hematopoietic stem cells, sometimes we call them hemocytoblasts. As you can see, they have big nucleus. They have lots of DNA materials. They have less cytoplasm. They have less proteins, as you can tell. As a general rule, these cells are destined to divide, destined for division. So for that reason, they have a much bigger nucleus than other cells. Of course, from these multipotent hematopoietic cells, as we saw earlier, we make common myeloid progenitor cells, and we make common lymphoid progenitor cells. From the myeloid progenitors, we're going to make our myeloblasts, mast cells, erythrocytes, and megakaryocytes. And of course, these megakaryocytes, look at it, how big they are. They can make our thrombocytes or platelets. Myeloblasts are going to make for us the basophils, the neutrophils. Look at how the neutrophils are multinucleated. Also look at to see how the basophils, they have all of these vesicles loaded with enzymes, as we're going to see later. Then we have the eosinophils. Look at that pinkish sort of coloration that they have. Finally, look at the monocytes and lock in the information about how the nucleus appears, which is sort of a bean-shaped. And of course, in the tissues, they turn into macrophages. And again, these are big big cells. Look at those vacuoles inside them, and look at those sort of purplish dots, that those are the vesicles that include the lysozymes and enzymes. Then we look at the common lymphoid progenitor cells, that they turn into natural killer cells. These are large granular lymphocytes, and they have lots of lysozymes. They have a big nucleus, they are big in size. And then we have a smaller size lymphocytes that they turn into T lymphocytes and B lymphocytes, and in the tissues, B lymphocytes make plasma cells. Again, look at how big is the nucleus of T and B lymphocytes. Let's start with this good starting question. Cells of innate system, neutrophils, macrophages, dendritic cells, natural killer cells, they have receptors that can identify broad classes of antigens and patterns that are common to the classes of the pathogens. For instance, they have receptors for identifying viral RNA, bacterial lipopolysaccharide, bacterial peptidoglycans, etc. Proteins that are required for formation of these receptors are encoded by genes that neither shuffle nor recombine, and they are fixed throughout the lifetime of the innate cells. What is the term that best describes these receptors? I've already covered this, and as you can tell, I'm drilling you a little before I go forward. These are germline encoded receptors. Lock in this information. These receptors or genes for these receptors are invariant. They are fixed. They have a low degree of inter-individual polymorphism, and they recognize conserved microbial molecular signatures, molecular patterns, or motifs. So they just look at how the intruders into the body look like. They have a general idea. This looks like a gram-negative bacteria. That bacteria that has intruded the body through a lesion on the skin looks like a gram-positive cocci. These patterns, again, just to familiarize your eyes and ears, motifs or patterns are general sort of appearances of the pathogens. Okay, let's look more closely and more specifically at the innate immune system. Let's first and foremost talk about innate immune response. Again, general patterns or motifs of invading pathogens are recognized by receptors of phagocytic cells. And all phagocytic cells have similar receptors, and they identify the very same general patterns. Recognition of pathogenic motifs and binding of receptors trigger activation of innate cells, such as macrophages, and causes engulfment of the pathogens. Activated macrophages also secrete chemokines, and these chemokines, as we're going to see shortly, they can attract more neutrophils, and they can attract monocytes from the bloodstream to the site of invasion by the pathogens. Neutrophils account for 60% of circulating leukocytes, and they are the first cells that arrive at the site of inflammation. So neutrophils account for 60% of circulating leukocytes. By far, this is the most prominent leukocytes in the bloodstream. They are the first cells that arrive at the site of inflammation because they are relatively smaller and they can get there quicker. The chemokines and cytokines produced by the innate cells, they can initiate a set of vigorous reactions, the so-called inflammation, that may potentially adversely affect the host tissues. Of course, it is good that they hurt the pathogens, but they may also hurt the host. That's why the site of inflammation may be painful, may be edematous. These are the issues that we're going to be covering later. Inflammation that results from release of cytokines is classically described by calor, means heat and fever, dolor means pain, rubor means redness and hyperemia, and tumor, which means the swelling. Let us look to see how pathogens induce inflammatory response. Of course, they do so by activating, first and foremost, the innate immune system. In this diagram, I'm showing the cells of the skin diagrammatically in a cartoon fashion, and I'm showing the bacteria. They look more like a bacilli in this case, but they are bacteria. And then I'm showing a macrophage and a dendritic cell. look at the macrophages, they are big cells in the tissues, and look at that unique dendritic cells. I'm just showing a few sort of arms, if you wish, or extensions on the surface. But they have many of these extensions, or arms, if you wish, on their surface. As you can tell, and as we're going to see shortly, these hair-like projections that they have increase their surface volume, and it maximizes their ability for identifying any antigen which is in a tissue. So they have the ability to identify antigens that are in the tissues. Even in a small amount, these cells can identify them and can grab them by those hair-like extensions. So let's go through this together. Pathogens invading the body are recognized by macrophages and surface receptors that recognize common, non-specific pathogenic motifs. Binding of the receptors to pathogenic motifs causes phagocytosis of the pathogens by these cells. So the macrophages and dendritic cells can phagocytize these pathogens. After phagocytosis, they initiate secretion of cytokines and chemokines. And those are the ones that allow communication of these macrophages and dendritic cells with other innate or adaptive cells of the body. Chemokines attract more neutrophils and monocytes from the bloodstream into the site of intrusion. The cytokines and chemokines also initiate inflammation that, among other things, activate proteolytic cascade of complements after recognition by the Fc receptors of the macrophages. Note that in addition to macrophages, the Fc receptor or Fc receptor protein is present also on the B lymphocytes, also on dendritic cells, also on natural killer cells, macrophages, neutrophils, eosinophils, basophils, human platelets, and mast cells. So all of these cells express Fc receptors, and this contributes to the protective functions of immune system in general. Innate response now activates adaptive response. So innate cells, they are the link between the site of intrusion of the pathogens and the adaptive immune system. Macrophages that phagocytize pathogens then get activated, in turn activate and present the pathogen to T lymphocytes. This action is in particular performed by dendritic cells that are specialized antigen presenting macrophages or antigen presenting cells. In these two diagrams, I'm showing that the macrophages, they have these Fc receptors on their surface. That's where the antibody attaches to these Fc receptors. And then the antibodies that I'm showing here, they have a binding site for the pathogen. The antibodies then can grab the microbes or the pathogens. And then these macrophages are gonna internalize the bacteria and do phagocytosis. In the lower diagram, I'm showing a dendritic cell under the skin, and after intrusion of the bacteria or pathogens, these dendritic cells are migrating to the nearby lymph nodes, and over there they present the pathogen to the T cells and B cells. Essentially, they introduce the pathogen to the adaptive immune system. Now let's see what I have in writing here. Like all other cells of the innate and adaptive immune system, dendritic cells in the bone marrow are developed from the same common myeloid progenitor cells that make neutrophils and macrophages. However, some experts also believe that dendritic cells, because of their linking position between innate and adaptive system, may also be generated from the common lymphoid progenitor cells that make the B cells, T cells, and natural killer cells. So, To summarize, the origin of the dendritic cells is questionable. The dendritic cells are claimed by some experts to belong to both innate and adaptive systems. After encountering any specific pathogen in the peripheral tissues, the dendritic cells get activated. They turn into potent antigen-presenting cells. They carry the information about the antigen to the nearby lymph nodes and pass it, or if you wish, present it to the cells of the adaptive system, such as T cells. And this is something that I showed in a lower diagram to you. What immunologic story does this picture tell you? I see a toll and I see a fly. This is toll-like receptor. These are molecules that alert the immune system to the presence of microbial infections. They are named for their similarity to toll, a receptor, first identified in a fruit fly, Drosophila melanogaster. Just to know, if you want to enter into a highway, you have to pass a toll first. That's why we have named these toll-like receptors. Just to know, in 1996, toll was found by Jules Hoffman and his colleagues to have an essential role in flies' immunity to fungal infections. And of course, that one is of historical perspective and value to us. In order to understand the concept of toll-like receptors, I have this question for you. What is the term that best describes membrane-spanning receptors on sentinel cells? Sentinel cells are first responders, such as macrophages and dendritic cells, that recognize structurally conservative molecules of the microbes when they breach physical barriers and activate immune cell responses. Of course, I know that you know the answer. These are toll-like receptors, TLRSs. And again, sentinel, if you didn't know, is a term that refers to first-line defense cells, such as macrophages. So let's talk a little bit more specifically about toll-like receptors. Toll-like receptors play a critical role in early innate responses. Receptors recognize only highly conserved structural motifs. As we said earlier, this pathogen-associated microbial patterns, PAMPs, that are solely expressed by microbial pathogens. Stimulation of toll-like receptors by these motifs starts intracellular events that ultimately produce a variety of cellular responses, including production of interferons, pro-inflammatory cytokines, and effector cytokines. For instance, when we stimulate TLR2 receptors, toll-like receptors 2, a stimulation of toll-like receptors 2, as you see in this diagram, activates MyD88 protein via AP1 protein. AP stands for activator protein 1. And NF-kappa B. NF-kappa B stands for nuclear factor kappa light chain enhancer or activated B cells. So when NF-kappa B, AP1 protein, is activated, that activation results in nuclear transductions that leads to a variety of cellular responses. For instance, in this case, we're going to make pro-inflammatory cytokines, we're going to make tumor necrosis factor alpha, and interleukin 6. So, what are the most common PAMPs, or pathogen-associated microbial patterns? Of course, I'm sure that you can guess what the answers are. These are peptidoglycan of the gram-positive and also gram-negative box, lipopolysaccharides, gram-negative box, flagellum on bacteria such as enteric bacteria, such as Klebsiella, bacterial DNA, and viral double-stranded RNA. So let's talk about MyD88 gene a little. I know it came up and you have questions about it. I'll try to make it more exam relevant and relevant to the concepts that are highly tested on the exam. This gene, actually MyD88, stands for Myeloid Differentiation Primary Response Gene 88, if you have been curious what this acronym stands for. This provides instructions for making the MyD88 protein that is involved in signaling within immune cells. MyD88 protein acts as a connecting protein that receives signals from outside of the cells and passes it to proteins that relay signals inside the cells and into the nucleus of the cells. MyD88 relays signals from certain Toll-like receptors and interleukin-1 receptors that are involved in early immune response to pathogens. When stimulated, MyD88 activates other proteins such as nuclear factor kappa B that ultimately regulates the activity of multiple genes, including genes that control the body's immune response, inflammatory reactions, and also genes that help to protect cells from signals that cause apoptosis. Deficiency of MyD88 causes predisposition to severe infections. So this is an important fact that you need to know in preparation for your examination. And another high-yield fact, the best way for me to present it is in the form of a question for you. A particular mutation in MyD88 protein is seen in 90% of patients who are presented with a non-Hodgkin type lymphoma that is characterized by an abnormally high number of lymphoplasmacytic cells in the bone marrow. It is postulated that this defective MyD88 protein overstimulates the nuclear factor kappa B even without signals from outside the cells. This allows for survival of abnormal cells that should normally have undergone apoptosis. What is the name of this lymphoma? I'll give you a hint. These lymphoplasmacytic cells produce a high amount of IgM. Any ideas? Waldenstrom macroglobulinemia. As you can tell, Waldenstrom is a lymphoplasmacytic lymphoma, and it gets its name because the lymphoma cells have characteristics that is a borderline between lymphocytes and plasma cells. That's the origin of the term.